This lecture is going to focus on Chapter 6 material from your textbook. That chapter is about pedigree analyses, applications of those types of methods, and genetic testing. It sounds like a potpourri of material that doesn't really have much in common, but actually there's a common thread through all of these things. This is how we use human subjects to study human genetics, which is fairly unique in the field of genetics. No model organisms, just human studies. So we'll start off discussing what the trouble is with studying humans, why this poses such a challenge, and also why it's important to do what we can studying humans. Then we'll move on to pedigrees. We'll define them, tell you how to decipher them, tell you what kind of information they give. We'll talk about twin studies and adoption studies and how those play into the understanding of human genetics. We'll discuss genetic testing, define it, talk about what it is, why it's used, and get you to think about whether or not genetic testing is a good thing or a bad thing. And then I'm going to end by introducing you to a career path called genetic counseling, which I think some of you might be interested in. So with a few exceptions, and don't tell Dr. Semperbond this, but with a few exceptions, most of the scientific research done in the life sciences is done either directly or indirectly for the betterment of man. There's plenty of research being done that is exploratory research to increase our understanding in general. And yes, that's very valuable research to do. It's extremely important for us to understand our environment, the history of our planet, and it gives us a much better idea of where things might be going. But within the hard life sciences, biology, genetics, etc., we're really studying what we're studying in order to better our own lives. As human beings, we want to live forever, or at least we want to live as long as possible and as healthy as possible. So we continue to do life sciences research in order to further our life quality and our life expectancy. We made the point in that very first lecture, but we have these model organisms, yeast, flies, pea plants, mice, bacteria, worms, you name it. But we use those to study humans better. Those are model organisms, and what they're modeling are human cells and human organisms. We are really always interested in how we work, but we use these lower life forms to increase that understanding. That's all fine and good, but if we're going to be studying humans and understanding humans, we need to look at humans directly too. And so pedigrees, twin studies, and adoption studies are all techniques that scientists use which allow us to study human beings directly without having to face impossible ethical hurdles. You see, people don't really like the idea of controlled crosses with human beings, test crosses and reciprocal crosses with human beings. Human beings are picky. They like things like love and marriage and free choice and free will, and that really gets in the way of well-controlled scientific studies. In addition, people have a very long generation time. It would take a lifetime to study a person's lifetime. That's really not so funny. But yeah, it takes about a lifetime in order for a human lifetime to go by. If we take the rule of thumb that it's about 20 years per generation, there's about 20 years separating human generations. That means to go from P generation to F1 is 20 years, right? It takes 20 years for someone to reach sexual maturity to a point where they should be a parent. Minimum. And then it takes another 20 years for the F2. So we're looking at a 40-year lifespan, a 40-year experiment just to get to an F2 generation. It's impossible. Also, people have very low ends, right? We usually have offspring numbers of 1, maybe 2. Triplets are rare, and anything above and beyond that is exceedingly rare. That gives us poor statistics for human studies. We talked about dihybrid crosses in an earlier lecture. If we have a dihybrid cross with heterozygotes for 2 traits, 2 genes, we expect to see that 9:3 to 3:1 ratio. 9 out of 16ths dominant for both traits, 3 out of 16ths dominant for the first, recessive for the second, 3 out of 16ths recessive for the first, dominant for the second, 1 out of 16th recessive for both. It's our hallmark ratio. To see that ratio, at a minimum, you need 16 offspring, 16 progeny, to see 9 of them of one, 3 of the next 3, and then 1. So if you have a family with 3 kids, how the hell are you going to see a 9:3 to 3:1 ratio? It's impossible. It's impossible. And finally, environment plays a huge role in human development. We've covered this already a few times when we've talked about nature versus nurture. So this makes human studies really, really hard. Everything we look for in high-quality scientific studies are lacking when we look at human beings. But we do have some tricks. fraternal twins versus identical twins, adoptions, and the detailed accounting of family records in specific families does allow us to sort out a surprisingly vast amount of data regarding human genetics. And so we're going to talk today about what we've learned about human genetics by studying human beings in the ways that we ethically and experimentally can. And we'll start with pedigrees. Most of you are probably familiar with the idea of a family tree in general. Family trees track family relations and family progressions across generations. Here is a family tree that's very near and dear to my heart. We have the Simpsons here. Bart, Lisa, and Maggie are all the children of Homer and Marge. Homer has his own brother Herb, who made an appearance, I think, in season 4. And Homer's father is Abraham and his mother is Mona. That's the paternal lineage of the Simpsons family. Marge's father was Clancy, mother was Jackie. She has her twin sisters, Patty and Selma. Patty and Selma have an adopted child, Ling. So we see this family tree as we expect to. Pedigree is nothing more than a genetic family tree. It's a family tree that considers the family relatedness, but with regards only to a single inherited trait. So this is a very simple pedigree that we see here. Give you a few ground rules for pedigrees. Pedigrees have males shown as squares, and we talked about that in the previous lecture with cytoplasmic inheritance. In pedigrees, females are represented as circles. Filled-in circles show the trait of interest, whether it be a disease or condition or just a trait, blonde hair, let's say. And non-filled-in shapes show the absence of the trait being considered. So by studying these inheritance patterns of traits represented on this family tree diagram called a pedigree, a trained geneticist can easily determine whether a trait is dominant or recessive, whether it's sex-linked or autosomal, whether it's polygenic or monogenic, in other words, governed by one gene or many genes, etc. And, believe it or not, by the end of today's lecture, you guys will be able to do the same and look at these pedigrees, and just by looking at them, have a very good sense of what type of trait is being shown. But before we can get into any of that, let's start with what these symbols mean. I already told you, but squares are male and circles are female. Filled in is affected, showing the trait we're interested in. Non-filled in is not affective, not showing the trait. And if you're using a trait like hair color or eye color, you have to pick what version of the trait the family tree is representing. In other words, it has to be one trait. So hair color is blonde. This pedigree shows blonde. That means everybody who's filled in on that pedigree has blonde hair. And everyone who's not filled in doesn't have blonde hair. Well, what about brown hair? I want a pedigree on brunette. Well, you can't use this pedigree for brunettes. You're going to have to design your whole new pedigree for brunettes because this pedigree is showing blonde hair. So it's truly a single trait, single version of a trait. Some other representations or denotions of pedigrees. A horizontal line, a line across a male and a female, show that they have mated and had children. Children from that mating are shown connected to a mating line, drawn vertically to the drawn perpendicular or vertically to the line across the parents. And those children are shown in their birth order left to right, with the oldest on the left and the youngest to the right. So siblings are connected horizontally. What I'm saying is shown right here. Let's get my trusty pen out. So if we draw your attention right here to this pedigree, this male and this female have mated. There's a horizontal line connecting them. Coming down perpendicular from that horizontal line is a vertical line connecting their children. This son, I know he's a son because he's a square, was their first child. They have a middle daughter and a youngest daughter. That's what we're saying here for these pedigrees. Each generation in the pedigree is represented by a Roman numeral, generation 1, generation 2, generation 3, etc. Each individual of a generation gets their own number, 1, 2, 3. If these children had cousins, they would be numbered 4, 5, 6 across the generational line. So person Roman numeral 3, number 8, is the 8th person of the 3rd generation of that family. And a slash through an individual means that they are now dead, they're deceased. You can see there are other denotions represented on this table from your textbook. If we have a colored dot in the center of the shape, it means the person is a carrier. They have the allele for the trait, but if the allele is recessive, they're not showing the trait. We have other ways of denoting things as well that I'm actually not going to get into on this slide. I don't want to overcomplicate it. So how do you analyze a pedigree? Well, here's a pedigree. It's a pedigree for a syndrome called Waardenburg syndrome, a very rare condition. Individuals that have Waardenburg syndrome are usually deaf. They have very poor vision, very fair complexion, and interestingly, a white forelock, as this kid shows here. So there's even a hair color condition that makes this Waardenburg syndrome very, very easy to diagnose. Do you think this is a sex-linked trait? Take a look at this pedigree. Now, I haven't taught you how to analyze pedigrees at all. Take a look at this pedigree and tell me if it's sex-linked, and think about why it might not be sex-linked. Think about what we said about sex-linked traits, what we noticed about the inheritance of sex-linked traits, etc. Well, it's not sex-linked. It's not sex-linked because sex-linked traits are usually passed from mothers who don't show the trait but have the recessive allele to sons who don't have another X chromosome and therefore can't mask that recessive allele. And if we look at this, we see that, first of all, women are showing the trait pretty prevalently, just as prevalently as men. X-linked traits don't show that. It's very, very rare for a woman to show an X-linked trait because she has two X chromosomes. In addition, we see instances where mothers have passed that trait on to both sons and daughters. And it is extremely rare for a mother to pass a trait, an X-linked trait, on to her daughter. Lastly, though, and most importantly, we see a mother not showing that trait. A father does show the trait, and the father has passed it to sons. That never happens with X-linked traits. Fathers pass Y chromosomes on to their sons, and traits that are X-linked never get passed from father to offspring, son or daughter. So this is not an X-linked trait. Do you think this trait is dominant or recessive? Take a look at this. Do you think it's dominant or recessive? That's a little bit harder to see at first blush, especially without talking about pedigrees, but I'll tell you right now that this is a dominant trait. Recessive traits are fairly rare because they're easily masked. And so to see recessive traits, you either have to have two recessive parents, both mating and having recessive children, or recessive traits are skipping generations because they are coming out by heterozygous matings. This Waardenburg syndrome is actually shown in each generation. This mother had it, and she passed it to both her kids. See, the father didn't have it. The father is dead now, but the father didn't have it. But still, the mother had it, and she passed it to both her kids. That seems pretty dominant to me. In this mating, one parent has it, and 1, 2, 3, 4, 5 children. 2 of those children, or roughly half, show the trait. Sounds like she's heterozygotic. This father has it, gave it to 2 out of his 3 kids. This father had it, gave it to 2 out of his 3 kids. So it's being passed from parent to offspring each generation. That sounds pretty dominant. So even without any formal training, even without any formal analysis of this pedigree, certainly no statistical analyses, we can spot some basic inheritance patterns. Not sex-linked, equally present in genders of a single family, not skipping a generation, probably dominant because parents are passing it directly to their offspring. Pick out all this interesting information in the absence of true Mendelian ratios and controlled crosses. So what we're going to do now is we're going to go into each type of pedigree, knowing the type of trait that it's showing, and we're going to look at the hallmark features of those pedigrees given those types of traits. So we're going to start with autosomal recessive, a recessive trait that can be masked by a dominant allele present on the autosomal chromosome. So here's a perfect example of an autosomal recessive pedigree, a pedigree showing an autosomal recessive trait. So what do we see? Well, most autosomal traits, whether they're dominant or recessive, should appear with equal frequency in males and females. And that seems to be the case here. One female has it, one male has it. So we see that. Sex-linked traits don't show equal prevalence in both genders. Sex-linked traits are much more common in males. Now, yes, we talked about sex-influenced traits and sex-limited traits. Yes, these traits show a bias given gender, but they're very, very rare. And so rare, in fact, that it would be unfair of me to ever give you a sex-influenced or sex-limited pedigree on any quiz or exam. So don't worry about that. If you see roughly equal prevalence of a trait, males and females, it is not sex-linked. And if you see a bias towards males, it's going to be X-linked. In this case, we're looking at autosomal recessive, so recessive traits should be pretty infrequent because they're recessive. They're easily masked. You need 2 of the alleles to show the trait, whereas you only need 1 dominant allele to show a dominant trait. So they should be pretty infrequent. They should skip generations because we do get masking by dominant alleles. Recessive individuals, when they're shown, should represent about 25% of a progeny, roughly speaking. Why? Because to get a recessive individual from 2 non-recessive parents, the only way that's possible is if both of the non-recessive parents were carriers, or in other words, were heterozygotes. And if both parents are not showing the trait, but pass it on to their children, thus both parents are heterozygotic, then we know from a heterozygotic by heterozygotic cross that only 25% of the offspring will show the trait. We know that. That should be second nature to us. So in general, if 2 non-trait showing parents have a trait showing individual, these 2 parents are by definition heterozygotic. It's the only way that's possible. It's the only way that's possible. And so this recessive individual should make up roughly 1/4 of the offspring. Now these parents didn't have 4 kids because they're humans and they have a poor N. But 1/3 of their kids, close enough to 1/4, excuse me, for me, are showing the trait. So another thing to look for. Inbreeding, the fancy name for inbreeding is consanguinity, or of the same blood, may often cause recessive traits to express. In this case, inbreeding has occurred between these 2 cousins. These 2 cousins have mated, and I can hear your ew from all the way over here on the other side of Pedigree, but these 2 cousins have mated, and they've had 4 kids, and one of those children is showing the recessive trait, because they're getting that recessive allele from their family lineage. And so there's a higher proportion or a higher chance of that recessive allele being in this one family than if they mated outside of the family. So inbreeding tends to lead to the recessive trait. Heterozygotes are more common within that affected family because that family is carrying that recessive allele. Breeding outside the family tends to bring in homozygotic dominant people, so breeding outside the family rarely results in recessive individuals showing the trait. So on to autosomal dominant traits. Well, being autosomal, we should still see that relatively equal frequency in both genders, so males and females roughly should show it in equal proportions. But since it's dominant, we do not see the skipping of a generation. Instead, we see that the dominant trait should be carried in every generation. Now, if this trait is dominant, we know that dad must be homozygotic recessive. It's the only way he doesn't show the trait. Now, mom could either be homozygotic dominant, and if that's the case, all of her children should show the trait, or mom is heterozygotic, in which case half of her children should show the trait. So by looking at this family tree right here, you should automatically be able to tell me with absolute certainty the genotypes of both of these parents in generation 1. Dad must be homozygotic recessive. That's the only way he's not showing the trait. And mom must be heterozygotic because 2 of her kids don't show the trait while the other 2 do. So we see that the proportions or ratios we would expect given the Punnett square hold true. Really, really interesting stuff. If an affected person mates with a recessive person, 50% of their children should be affected, assuming the affected person is heterozygotic. And that is, in fact, what we see in all of these generations. Roughly 50% of the offspring are carrying the trait and 50% aren't. 2 recessive parents will never have children expressing the trait if the trait is autosomal dominant. Because if 2 recessive parents, 2 parents not showing the trait, are mating, that means they're both homozygotic recessive. That means there is no dominant allele to pass on. And so 2 non-expressing recessive parents will never, ever have children that show the recessive trait. And we see that here as well. Familial give me a second here. Let me start that again. Familial hypercholesterolemia, I think I added an extra syllable there, is an autosomal dominant trait. That is the pedigree we're looking at here. That means that individuals only need one allele to show this trait. This causes a body making too much cholesterol for itself. And just as a side point, this is the condition that statins, prescribable statins, fix. If you're eating a very high cholesterol diet and your blood cholesterol is high and your doctor puts you on a statin, he or she has no idea what the hell they're doing, and you should go seek another doctor at your earliest convenience. The drugs that are marketed for high cholesterol have no effect whatsoever on dietary cholesterol, period. They only stop the body from making too much cholesterol. They only help people that have a genetic disorder of hypercholesterol, blah, 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 blah. So it's just something to know for your own personal interest. All right, so moving on. What does an X-linked recessive family tree look like? Something like colorblindness, for example. These traits are easy to spot. First of all, they're X-linked, so they are not found in equal prevalence across both genders. Males should be almost exclusively affected for the genetic reasons we've already discussed when we talked about X-linked traits. These traits tend to skip a generation. Why? Because they're recessive, and recessive traits always tend to skip a generation on a lineage. Now, let me emphasize that point, on a lineage. So you look at this pedigree and you say, well, wait a minute, Dr. M, this doesn't skip a generation. See, we have an affected individual in generation 2, an affected individual in generation 3, and an affected individual in generation 4. So it's not skipping a generation. But I mean skipping a generation on the lineage. Here, neither parent has it. This kid does. Next generation on this lineage, no one has it, and then someone does. Here we have, yes, we skipped 2 generations, but then the trait pops up. The next generation, no one has it. It's quite likely in generation 5 that we will see an affected individual. So when we suspect that a woman is a carrier, and we can suspect that because if the father or husband does not have the trait, then we know he's not carrying the allele. Remember, this is X-linked. So if he had the allele, he would show the trait. He's only got one X chromosome. So if we have a child that is showing the trait, especially a son, if we have a son showing the trait, and the father is not showing the trait, we know that that trait came from the mother. The mother is the only person who gives that son his X chromosome. So when a woman is suspected of being a carrier, she's heterozygotic, she has a normal allele, but she's also carrying the recessive allele. Approximately half her sons should be affected, but none of her daughters. Because if her husband is not showing the trait, he is giving a dominant X chromosome, a dominant allele, and all the daughters should be protected. We then denote the mother with a dot in her shape because we know she's a carrier for the trait. X-linked recessive traits are never passed from father to son because fathers don't give sons X chromosomes. Affected fathers will have carrier daughters, guaranteed. So this father is colorblind. He shows the trait. Every single one of his daughters is a known carrier now. Because they got one dominant allele from their mother, that's why they are not colorblind. But the one and only X chromosome that their father had to give them was carrying the recessive allele. So this woman now risks, if you want to call it a risk, giving her sons or half of her sons that recessive trait. We know that. We can watch it in the pedigree. Finally, in the rare case of an affected mother, so this is possible, women can be colorblind, women can have X-linked traits, they have an affected father that gave them a recessive allele on the one X chromosome he donated, and they have a carrier mother who gave a recessive allele on the X chromosome she donated, so then the woman is homozygotic recessive. In the rare case of an affected mother, all of her sons will have the trait. 100% of her sons will have the trait, because regardless of which X chromosome she gives to her son, she is passing the trait on to them. Hemophilia A is an X-linked recessive trait. And the Bleeders, the royal family, the Bleeders, show this in their pedigree. Now, the wonderful thing about these royal families and other such historical families is that the family records are amazingly detailed. Because the family lineage matters so much in these families, they take meticulous care and notes when it comes to all relations among the family. And then if there's a condition, that condition is tracked as well, and you can assemble a pedigree from the family tree and the history of the conditions. So this is the pedigree for hemophilia in the royal family, and this type of hemophilia is X-linked. So think of the rules I told you for recognizing X-linked traits, and look at them here. No daughters affected, no women affected on this family tree at all. A very stark gender bias towards the males. So right away you have to think sex-linked, X-linked. Something's got to do with gender. Then, is it dominant or is it recessive? Well, if it were dominant, we would see it in every single generation across lineages. If it's recessive, it should normally skip generations on a lineage at least, if not skip multiple generations. And we see here, especially on this lineage, no presence of the trait, a son had it. No presence of the trait, a son has it. No presence of the trait here, no presence of the trait on this lineage, two sons have it. No presence of the trait here, no presence of the trait here, but then on the lineage, these sons have it. So we are skipping generations. And it's clear to see that it's not present on every member of a lineage within each generation. Okay. What about X-linked dominant? There is such a thing as being an X-linked dominant trait. X-linked dominant traits are seen in both sexes, but they tend to affect females more. So if we see a female bias, we can start thinking X-linked dominant. Each affected individual should have one and at least one affected parent, because remember, we're talking about a dominant trait. So if two unaffected parents are mating, we know there are no dominant alleles among those parents. If they're unaffected, they're homozygotic recessive, or in the case of the father, his 1X chromosome is recessive. So non-affected parents should have nothing but unaffected children. Affected children should have at least one affected parent. These traits do not skip generations because they are dominant. But the best indicator for X-linked dominant traits, the one that will never fail you, is that affected fathers pass these traits on to all of their daughters and none of their sons. I'll say that again. An affected father will have all affected daughters and no affected sons. I would like you to think about that for a second and see if you can figure out why that is. Oh, I love this stuff. I just love this stuff. You can understand this. In the few brief weeks that we've been doing these genetics, you can get this. It's X-linked dominant. So here's Dad. Here's his X chromosome. Here's his Y chromosome. He shows the trait. And so we know that he is carrying that dominant allele. Capital D. Here's Mom. She has two X chromosomes. But she does not show this dominant trait. Therefore, she is homozygotic recessive. Now let's talk about the daughters of this mating. Daughters have to have two X chromosomes by definition. One of those X chromosomes came from Dad. And Dad only had one X chromosome to give. And that X chromosome had that dominant trait. Mom gave an X chromosome. Mom only has one type of X chromosome to give. And it's the one that shows the recessive trait. So all of the daughters are heterozygotic, but all of the daughters show the dominant trait. Bingo, bingo, bingo. What about the sons? Well, the sons get the Y chromosome from the dad. That's what made them sons. And the sons got an X chromosome from Mom, but Mom only had one type of X chromosome to give, and it was recessive. So none of the sons show the trait, because the trait comes from Dad, and Dad did not pass on an X chromosome. Bingo. Bingo. This is the hallmark feature of X-linked dominant, and it will never, ever fail you. And it's a beautiful indicator of how we can understand genetics at a very fine level. I just went over the why. Affected mothers are usually heterozygotic. So affected mothers are carrying one dominant allele and one recessive allele usually. And so they will typically pass that trait to half of their sons and half of their daughters. Let's see if we can find that right here. So this mother here is affected, but she's heterozygotic. And the father is not affected, so we know his genotype, his 1X chromosome, is recessive, because he's not showing the trait. Imagine this mating as a Punnett square. Half of the sons are going to get the dominant X chromosome from mom. Half of the sons are going to get the recessive X chromosome from mom. All of them will get the Y from dad, meaning half of the sons will show the trait, roughly on average, and half of the sons will not. Half of the daughters will get the dominant chromosome from mom. Half of the daughters will get the recessive X chromosome from mom. All of the daughters get the recessive X chromosome from dad. And so half of the daughters will show the trait, and half of them will not, roughly on average. Y-linked traits. There isn't really much going on the Y chromosome except for SRY. The Y chromosome is pretty useless. A Y chromosome inheritance pattern on a pedigree would look like this, theoretically. Theoretically, the pattern of Y inheritance would be a trait that is passed only from fathers to sons, never from mothers to sons, never from mothers to daughters. Why? Because fathers give their sons Y chromosomes. That's why. Never skipping generations, because there's always a Y chromosome to be had. Never passed from daughters or mothers to children. However, to date, the only Y-linked trait is being male. That is a trait. That is a phenotype, right? The gender is a trait of an organism, and so certainly the Y chromosome passes that trait, and we see this inheritance pattern, right? Males have male sons. That's passed from father to son. And women don't pass maleness because they don't have maleness to give. But really, that's all that goes on as far as the Y chromosome. So, this pedigree here represents the inheritance pattern of a rare disorder in an extended family. What is the most likely mode of inheritance for this disease? And assume full penetrance and all that good stuff. So what do you see? Well, what do you see in terms of gender? Is it equal numbers of males and females, or mostly males or mostly females? So if you said it's mostly males, you got to start thinking right away, X-linked recessive, X-linked recessive. X-linked dominant tends to affect females more, and anything autosomal tends to affect males and females equally. So we got to start thinking X-linked recessive. There's another hallmark feature of X-linked recessive, and that it skips a generation on the lineage. So do we see that? Well, they say, no, I don't, Dr. M. There it breaks down, because it's in generation 2, generation 3, and generation 4. And then I will remind you, intelligent student, that we don't mean the generation as a family, we mean on the lineage. So no prevalence of the trait here. No prevalence of the trait here, and then the trait crops up. Skips 2 generations, and then it shows. No prevalence of the trait here, but then the prevalence of the trait here. So it skipped a generation because it wasn't here. Here's the trait. Next generation on that lineage, no trait, then the trait crops up again. We skipped a generation. So yes, absolutely. We're skipping generations. This is an X-linked recessive trait. Table 6.1 in your textbook, perhaps one of the most helpful figures that I will recommend to you this semester, takes everything I just told you and breaks it down into these kind of thumbnail pictures. What to look for autosomal recessive on a pedigree. What to look for autosomal dominant. What to look for X-linked recessive. What to look for X-linked dominant, and even Y-linked, what to look for. So I call your attention to those tables in your textbook because they are your cheat sheets for deciphering pedigrees. On to twin studies. There are two types of twins. I'm certain that many of you know this already, but we'll just make sure we're all on the same page. Dizygotic twins, twins that arise from two zygotes, are more commonly referred to as fraternal twins. Monozygotic twins come from a single zygote, and we more commonly refer to them as identical twins. There is a stark difference between these two twin types. Dizygotic twins arise when a mother releases two separate eggs simultaneously, and each of those two eggs are fertilized by two independent sperm cells simultaneously. Therefore, dizygotic twins are no more related on the DNA level than standard siblings. That is, they share on average about 50% of their DNA content. So other than being born on the same day and being the same age, dizygotic twins are siblings. Monozygotic twins, however, arise when a mother releases a single egg. That egg is fertilized by a single sperm, giving rise to a single individual. But then very early on in development, that one cell, as it's dividing, separates into two separate cells and develops as two separate independent embryos. Because of this, because that all arose from a single fertilized cell, monozygotic twins are truly genetically identical. They are naturally occurring genetic clones. This diagram kind of shows what I just said. If you need that, you can pause it and take a look at this. So we can use this to our advantage. Fraternal twins are kind of like a control, an experimental control for environment, while identical twins are a type of experiment or control for the nature or genetic component of traits. Why? Why? Well, when we compare monozygotic twins and dizygotic twins, when we compare those for a trait, we can begin to understand and parse out how much of that trait is genetic and how much is environmental. Because dizygotic twins were born to the same family at the same time. They were raised in the same home by the same parents, probably fed the same food, probably went to the same school, probably had nearly identical environments. Monozygotic twins are born at the same time to the same family, probably had the same environment at home, probably went to the same school, probably drank the same water, probably ate the same food, but also had the same exact DNA. That's the only difference between dizygotic and monozygotic twins, is that DNA similarity, that DNA identity. So, if dizygotic twins show the same trait in the same way compared to one another, then that trait likely has a strong environmental component, because dizygotic twins are only 50% the same at their DNA level, unlike monozygotic twins. But dizygotic twins are usually close to 100% the same environmentally, because they're raised in the same family, same house, same water, same food, same. I'll give you an example. If I were to have dizygotic twins after shooting myself, because I can barely handle two kids that are different ages, if I had dizygotic twins, it's very likely, one would hope, that they would both grow up to be Bruins fans. Because they're being raised in a house that has a Bruins fan in it. They're raised in a house that has a Bruins logo Christmas ball hung on the tree, that has Bruins posters in the basement, that has a father who puts on Bruins games every time they're on TV. They wake up each morning getting a rundown over breakfast of how the Bruins did the night before, where the Bruins are in the standings. It's a pretty Bruins-heavy household. And so if those dizygotic twins grow up being Bruins fans, they're going to have a high level of similarity with one another. In other words, they're both Bruins fans. They're similar. But they're similar not because of their DNA. They're similar because of the house they were raised in, because of their environment. Monozygotic twins are much more alike genetically than dizygotic twins. So if monozygotic twins are more alike for a trait than their dizygotic counterparts, then that trait likely has a strong genetic component. In other words, if we're looking at a trait And we see that, about 75% of the time, dizygotic twins grow up being fans of the same sports teams. You say, well, okay, that makes sense. That makes sense because they have the same environment. And then you see 95% of the time, monozygotic twins grow up to be fans of the same sports team. You say, wait a minute then. As strange as it seems, why would monozygotic twins be more likely to follow the same sports team than what do monozygotic twins have that dizygotic twins don't? Genetic identity. And then you start saying, I wonder if being the fan of a sports team is genetic, as crazy as it sounds. Because why else would monozygotic twins be more alike than dizygotic twins for that trait? The only thing that monozygotic twins have that dizygotic twins don't is identical genomes. They both share the same environment. So you're probably wondering at this point, what the hell is he talking about? So we're going to run through some examples and try to drive the point home a little bit more simply. Concordance, the term concordance, is the measure of the percentage of twin pairs that have the same trait, whatever that trait is. So if the concordance for sports team fanship is 75%, we mean that 75% of twins are the fan of the same team. That's their concordance. If there's a higher concordance in monozygotic twins than in dizygotic twins, we are looking at a genetic component. Here's a table from your textbook showing concordance of both mono and dizygotic twins for several traits. So how do we read this? Well, trait 1, heart attack in males. The concordance for heart attack in males of monozygotic twins is 39. What that means is that 39% of the twins that were studied showed that if one twin had heart attacks in males, the other twin had it too. 61% of the twins were where one male had it, the other didn't. When one twin had it, the other didn't. Okay? 59% of twins, monozygotic twins, if one twin has epilepsy, the other twin had it too, 59% of the time. That's how you read this table. And then the same for dizygotic twins, it's just that they're dizygotic. So when we read this table for bronchial asthma, we see that 47% of the time, if one monozygotic identical twin has asthma, the other twin has it too, 47% of the time. That means 53% of the time when one twin has it, the other doesn't. But for dizygotic twins, 24% of the time when one twin has it, the other has it as well. So you say, wait a minute here. Dizygotic twins have the same exact environment. They're breathing the same foul air, if such is the case. They're living in the same home, the same town, eating the same foods. And only 24% of the time, if one dizygotic twin has asthma, the other has it too. But now we look at monozygotic twins. Same foul air, same home, same food, same parents, same environment. But we nearly double that concordance. What could possibly explain a higher concordance in monozygotic twins versus dizygotic twins? Can't be more similar environment. That doesn't work. It's got to be the genetics. It's got to be a genetic component. So in general, what you do is you take the monozygotic concordance, you subtract from it the dizygotic concordance, and if you're left with a pretty high number, you're looking at a genetic component. If you have any trouble with this, if this is still kind of inaccessible to you, if this is still abstract and you don't know what to make of it, please come see me. Put it in your reflection, of course. If the majority of the class struggles with this, we'll review it on Monday. But if you find yourself struggling with it, come see me. This is an important component of this lecture, something you should know. If any of you know of or have identical twins, you're aware of how similar they are in many ways, but also how different they are. And then you also have to ask yourself, well, are the environments of mono and dizygotic twins really comparable? I think they largely are, certainly in terms of food and home environment, love and affection and all that, but they're not 100% the same, right? Parents often dress monozygotic twins in the same exact outfits. They rarely do that for dizygotic twins. Parents often mix up monozygotic twins, especially when they're younger, giving them a very, very similar home environment. Whereas for dizygotic twins, there's often a lot of tailoring and individual nurturing going on. So there's flaws in the scientific method of this, yeah, but it's the best we can do with humans, right? So let's look at asthma a little bit closely. We all know what asthma is, I think. It's difficulty breathing, constriction of the airways, etc. Asthma rates in children is climbing at an alarming rate, and of course it makes sense to blame pollution and the poisoning of the earth and all of that. It makes sense. I mean, I feel the same way. That means it's an environmental cause. We're dumping all this crap into the air. Because of that, the air becomes foul, it becomes toxic, and it creates problems in our physiology. But how do we know? How do we know for sure it's environmental? Well, we do concordance studies. Scientists in the UK a few years back studied about 5,000 mono and dizygotic twins to look at concordance given in asthma. And if there's an environmental component, we should see a high concordance in dizygotic twins, but we should see no higher concordance in monozygotic twins. That's the hallmark of an environmental component. If we go back to our rule of thumb, back here, the subtraction value for these concordance studies should be 0 if we're looking at something that is completely environmental. Meaning, having a higher amount of DNA does nothing for you because the trait is completely environmental. So what? You have more DNA in common. If it's environmental, that doesn't affect the trait at all. So they did this study, and this is what they saw. They looked at a lot of twins, and they saw the concordance for dizygotic twins given asthma is 37% in this study. Okay, so 37% of twins, if one twin, of dizygotic twins, if one fraternal twin had asthma, the other had it too. So that's a pretty high percentage. That alone might lead you to think that it's environmental, because about 40% in the study, one twin has it, the other has it. They're sharing the environment. That makes sense. They're sharing the same environment. They share the same outcomes. But for monozygotic twins, the concordance was 65%. In other words, it was more likely for monozygotic twins, if one had it, the other would have it too, than for dizygotic twins. Well, the only thing monozygotic twins have that dizygotic twins don't is a higher degree of genetic similarity. So we can look at this two ways. There is a definite genetic component, because if we subtract the dizygotic number from the monozygotic, we're left with a pretty high number, 28 in this case. But environment plays a role too, because if it was purely genetic, if bronchial asthma was 100% genetically based, the concordance in monozygotic twins would be 100%. And it's not. So there must be something above and beyond genetics, because there are 35% of monozygotic twins that where one twin has it, the other twin doesn't. And if it was purely genetically based and they have 100% genetic identity between each other, why is that 35% discrepancy there? So it's a little bit of both. So here's a concept check for you. Higher concordance, well, you know all that. So the question is, a trait exhibits 100% concordance for both monozygotic and dizygotic twins. What conclusion can you draw about the role of genetic factors in determining differences in the trait? Okay, I want you to think about this. I want you to answer this honestly for yourself, and then go to your textbook. And in your textbook, at the end of the chapter, you can find the answer to this question and see if you're right or see if you're wrong. And if you're wrong, think about why, and maybe put that in your reflection or bring that to class. Make that something you and I talk about, either in class or outside of class. What we can glean from adoption studies is a little bit more intuitive, not nearly as abstract as pedigrees or twin studies. But we use similar logic. We can study adoptive families, compare them to biological or traditional families, and we can glean quite a bit of genetic information from that. Let's consider obesity first. What would you think if I told you that obesity rates in adopted children don't really correlate well with their adoptive parents? In other words, heavy adoptive parents don't necessarily have heavy adopted children, and heavy adopted children don't necessarily have heavy adoptive parents. Then I told you that obesity rates in adopted children do correlate well with their biological parents. In other words, heavy biological parents often have heavy children, even when those heavy parents gave those children up for adoption when they were infants, never met those children, never raised those children, never influenced those children's eating habits, etc. Well, that's really the truth. That's how it works. Here is a chart that shows biological parents, their BMI, the mass index of the parents, the body mass index of the parents, over the weight class of the adopted child. Okay, so it's the biological parents with their child. When the biological mother is thin, the child tends to be thin. When the biological mother is heavy, the child tends to be heavy. When the biological father has a low BMI, the child tends to be thin. When the biological father has a high BMI, the child tends to be heavy. Interesting. Now for the adoptive parents. Same breakdown, same chart. Fathers with relatively low BMIs, or higher BMIs have children that are thin or heavy. It makes no difference. Mothers with relatively low BMIs, or high BMIs have children that are thin or heavy. It makes no difference. So we can conclude from this, as surprising as it might be, that environment really doesn't have the role in obesity we would expect it to. It's not children learning bad habits from their parents. It is children inheriting metabolic predispositions. And I dare say children inheriting behavioral preconditions from their biological parents that affects their eating habits and their weight gain. From a genetic and DNA perspective, adoptive children and their adoptive parents are genetic strangers. They have no DNA similarity between them whatsoever. From a genetic and DNA perspective, biological children and their biological parents, even if social strangers and having never met, have 50% identity between their DNA. So if adopted children resemble their adoptive parents more, and they are genetic strangers, that trait probably has a large environmental component. Families typically share the same environment. But if adopted children represent their biological parents more, such as for obesity, but the same is true for schizophrenia and many other traits, that trait probably has a large genetic component. So we can tease out genetics and environment by using these social situations like adoption, using these family conditions like twins to our advantage. Genetic testing. Between pedigrees, twin studies, adoption studies, basic research using model organisms, and the sequencing of the human genome, we now know a whole hell of a lot about the genes that cause traits and disease in our genome. So what do we do with all of that? Well, one thing we do more and more of is in utero and infant genetic screening. We call this genetic testing. The idea behind this is if we can spot a genetic disorder early on, perhaps we can intervene with some treatment or therapy to alleviate some, if not all, of that condition. We use some things, such as ultrasonography, to provide a visual inspection of the developing fetus in utero. This is an ultrasound of my older daughter, Charlotte. You can see my wife's name up there in the upper left-hand corner, the date this ultrasound was done. This was amazing to us, actually, because if you see, just by chance, the tech caught it just right. You see my daughter has her arms up at her sides, so her hands are touching her cheek here, and you can actually see the bones of her arm. So the ultrasound has penetrated her soft tissue to her bone there, but because of the depth, the field, it actually was just able to scratch the surface of her face without penetrating her skin. So you actually see my daughter's face in this ultrasound. And my daughter was born in May. And if you can see, this ultrasound was done in November. So we're talking about roughly the 4-month time frame, still very, very small, very, very early on. And we were able to see a cute little nose there of our first daughter. So you can do these visual inspections. Some conditions do present visually. Even if they're genetically based, they can do so in utero. Spinal bifida is an example. Spinal bifida is not genetically based. I don't want to be misunderstood there. Spinal bifida is due to a folic acid deficiency as embryos developing in utero, but certain things can be spotted with physical features with ultrasounds, and spinal bifida is an example of that. Amniocentesis. This is the insertion of a long needle directly through the belly into the amniotic sac to withdraw amniotic fluid. Floating in the amniotic fluid are shed fetal cells, either from the placenta or from the fetus itself, and those fetal cells can be extracted, DNA can be gotten from them, that DNA can be analyzed, and genetic disorders can be recognized with that. Chorionic villus sampling, or CVS, goes in through the vagina to and through the cervix and actually steals some cells from the placenta through suction, without the use of a needle, and then these placental cells are tested for genetic disorders. You may or may not know this, but the placenta is actually a genetic clone of the fetus early on in development. Some embryonic cells differentiate and become placenta. So placental cells are genetically identical to the fetus. If you can get placental cells, you can measure the chromosomes, the genotypes of the fetus. And now there are some maternal blood tests available that purport to detect fetal conditions. So you can just take blood from the mother. It's a typical blood test for the mother, and they offer some type of predictive value for what might be wrong for the fetus. These come with fairly high positive rates. I don't know why any doctor in good conscience would ever recommend such a test, but all of them do. So the test comes back positive. The positive result is untrustworthy because there's a high false positive rate for the test, and so you make the parents worry for the next 4 months for something that might not be an issue in the first place. But it's done, and these tests are recommended for pregnant mothers. And believe it or not, a small number of fetal cells, fetal stem cells, have been shown to circulate in the mother's bloodstream. They detach from the placenta, they get into the maternal bloodstream, and they circulate harmlessly in the maternal bloodstream. If these cells can be captured from the mother's blood and tested, we would have a relatively risk-free fetal DNA test using nothing more than a maternal blood draw. Techniques for doing this are being developed as we speak, but it's still not successful enough to be used therapeutically. Things like amniocentesis and chronic villus sampling, these things do have inherent risks. The fetus can be harmed, spontaneous abortion can be triggered, and so these tests should all be done with those risks in mind. IVF is more and more common now, in vitro fertilization, this test tube baby cloning stuff, especially for parents who have difficulty conceiving in the traditional way. And because these zygotes, these fetuses, are being fertilized in vitro outside of the body, they can be easily tested for genetic disorders prior to implantation in the mother's womb. And so pre-implantation genetic diagnoses means testing these fertilized eggs prior to implantation, selecting ostensibly those that have no genetic conditions or disorders, but perhaps those that have the preferred traits of the parents, such as the preferred hair color or the preferred eye color, etc. Interesting. But, of course, we could check for things that are easy to see on genetic testing, like trisomy 21, which is Down syndrome, and any IVF embryo that had trisomy 21 would not be implanted to avoid the birth of a child with Downs in that scenario. And now there's also personal genome testing for all of us. You and I can go to a pharmacy, buy an over-the-counter genetic test, take a little Q-tip swab from the test, swab the inside of our cheek, put it in a little test tube, ship that off to the company, and get genetic tests back for some fee. The societal question for all of us becomes, as budding geneticists, is this good? Should we be doing this? Should genetic testing be so available? Should we have this information? What does this information mean? What does this information do for us? Do we want pregnant mothers to know all this information? What will they do with this information? How it will impact their decisions? These are just things to think about. By law, believe it or not, by law, all newborns must be tested for an array of genetic disorders. These are the genetic disorders being tested for as recommended by the American College of Medical Genetics. All of these disorders are tested in every newborn child. This is without parental consent. This is without often parental knowledge. All of these things are being tested for. Now, sometimes this is good. I mean, here's phenylketonuria. We spoke about this already, PKU. You need to know that right away. If you can spot PKU right away by a genetic test, you can get that infant off of a phenylalanine high diet, and you can protect that fetus from mental retardation. You can intervene environmentally and protect the fetus. So I'm not against this testing. If you sense cynicism from me, I'm not against this testing. What I'm against is the lack of informed consent. Parents should be told that their infant is being tested for this, in my opinion. They should be told why that testing is being done, and they should be given the results of those tests. I have 2 young children. Both of those children underwent this screening. I know that because it's the law in Massachusetts that this be done, and my kids were both born in the state of Massachusetts. Yet I don't have the results of any of these genetic tests for my children. It's interesting. In addition, the infant fetal blood drawn for these tests, and it usually comes from the umbilical blood, so the baby is not punctured, for these blood draws at once born. So the umbilical blood serves this purpose, but that umbilical blood that was used for these tests is often kept by the state indefinitely, again, without parental consent or parental knowledge. So genetic counseling, what is this field, this career path called genetic counseling? Well, it ties in perfectly with everything we're saying here, and I think it's just something that very few people have heard about, and many, many people might be interested in. Parents-to-be, now, based on everything I told you, get bombarded with all of these things. Maternal blood tests for genetic disorders, amniocentesis, chronic villus sampling, all these options for tests, all these test results, etc. And it is overwhelming. It's so much data with so many disorder names that are barely pronounceable. Most parents have no idea what it all means. Because of the simultaneous explosion of the genetic information that's available to parents, a relatively uninformed lay public that knows very little about these disorders, and the rapid overtaking of medical doctors' time with just too many patients and too little time to meet with them, this relatively new career path of genetic counselors was born. Genetic counselors are genetic experts, and they partner between, or serve as the liaisons between, naive parents who are getting all this information and busy doctors that have no time to talk to those parents about these results. So the genetic counselor serves as the teacher, the explainer, and the advisor to parents regarding all genetic matters. And as genetic tests are becoming more mainstream even for adults, genetic counselors are beginning to work with adults about their own genetic health in addition to expectant parents. Genetic counselors are trained to provide non-directed information, meaning here's what the tests mean, and you need to decide what to do about those results, as well as directed information, such as here's what these tests mean, and you may want to consider changing your diet now as an intervention for your child, so you give some direction in addition to explanation to the parents. And genetic counselors are counselors, so they're also trained to deal with the psychological ramifications of these tests, the worrying and fretting, the panic that often falls upon expectant parents when they get negative information. Most genetic counselors have a 2-year master's degree. They're board certified by genetic counselor boards. There is the National Society of Genetic Counselors, and their website is www.nsgc.org, as well as the American Board of Genetic Counseling, which oversees the board certification process, www.abgc.net. And at both of these websites, you can not only find information about the career and a little bit more about what the average genetic counselor does, but more importantly, you can find where programs for genetic counselors are offered nationwide. So I just thought that's something some of you might be interested in. If you like biology and you think you like genetics, but you have no idea what the hell you want to be in 2 years when you all grow up, maybe this is something that'll interest you. So what was talked about in this lecture? We talked about pedigrees, twin studies, and adoption studies as being a workaround for dealing with human studies without the ethical hurdles that often come with human experiments and the experimental limitations that get in the way, such as ethical treatment of human beings, love and free will, low N, they eat too much, they don't like to stay in cages, and all that stuff that kind of gets in the way with humans. What is a pedigree? A pedigree is a family tree, nothing more. But it's a family tree with a genetic slant. It considers a trait, a single inherited trait, and nothing more. We went through the rules of thumb for these pedigrees. Autosomal traits appear with equal occurrence in both sexes on average. Recessive autosomal traits occur in both genders, but usually pretty infrequently, skipping generations. Dominant traits occur in both genders fairly equally, but don't skip generations. We talked about recessive X-linked traits are male preferred, almost exclusively male, but they skip generations. X-linked dominant had that hallmark feature that'll never let you down. Fathers pass this trait to all of their daughters and none of their sons. We talked about concordance and comparing monozygotic twins with dizygotic twins, and we can use this to parse out how much of a trait is from genes and genetics. If the dizygotic twins show the same trait in the same way, then that trait is likely environmental. If monozygotic twins have a higher concordance and they're more alike, then that trait also likely has a genetic component as well. We can use the same type of logic for adoption studies. Adopted children that resemble their adoptive parents more probably are showing an environmental component. Adopted children resembling their biological parents more are probably showing a genetic component. We talked a little bit, too, about genetic testing and what's done to newborn infants, as well as that idea of genetic counseling. So we're going to change gears pretty heavily now, and we're going to start talking about what happens when two genes for two different traits are on the same chromosome. That means they are physically linked to one another, and that changes the predictions and the patterns of inheritance radically. So we'll begin those topics in the next lecture, and I can't wait. I'm sure neither can you. Thanks for watching. Where's my cursor?